Good evening, everyone. We're at 7 o'clock. We'll just give it a couple of moments as we see people joining. I can see we've got about 20 people on the call, but I know we've got lots and lots of people registered, and the number is shooting up fast. So we'll just give it a moment or two. For those who don't know me, my name's Ed Holloway, and I'm the Chief Executive at the Macular Society. And this is my first time kind of co-hosting a My Macular and Me webinar. I was briefly introduced in Kathy's goodbye webinar back in the summer, but this is my first time on hosting duties. So a real pleasure to be with you all this evening, and hopefully I'll fill Kathy's shoes adequately. And I've got an old hand as well, Emma, joining me from the Macular Society to help out with hosting and also to help out with the Q&A session later. But the key people here today are Dr. Ashley Wood and Vera Silva from Cardiff University, who are here to discuss the team's development of technology to detect early changes in vision, especially difficulties adjusting to low light, which could help diagnose AMD sooner. So Dr. Wood is a senior lecturer at Cardiff University School of Optometry and Vision Sciences. He studies as an optometrist at Cardiff before returning to undertake postgraduate training in research. In addition to training future optometrists, he continues to pursue research in age-related macular degeneration as part of the university's macular research group. And this work involves the use of ocular imaging technologies such as OCT and imaging retinal densitometry, apologies, bit of a mouthful, to detect and monitor early AMD. And Vera Silva is a PhD student supervised by Ashley, Tom Margrain and Rhiannon Reynolds with the Cardiff University Macular Research Group. And her PhD project is funded by the Macular Society. I'm now going to hand over to Dr. Wood and Vera, but please do pose your questions for our speakers in the chat as they go through their talk, because we'll be asking those questions in the second half of that talk. And now I'll pass over to Dr. Wood. Thank you very much.
Thank you for the introduction, Ed. It's a pleasure to be here. Tonight, I'm planning to talk to you about imaging retinal densitometry and hopefully share a little bit of the understanding of why we've been developing this technology at Cardiff University, what the potential role it can play in the assessment of AMD and also the understanding of the condition. And so I will start off by taking us through that. And then I'm joined here by my PhD student, Vera Silva, who will be telling you a little bit about the use of this technology in her PhD project. So I just want to start off with a little bit, just talking a little bit about the Macular Research Group here at Cardiff University, because all of our research is not really individuals, it's a team effort. All those faces you can see displayed there are part of the group and conduct projects related to macular disease. The top row there, obviously you can see myself and Vera. And in fact, the next person along is Professor Tom Margrain. Some of you have been to events like this before, might have come across his name. So really, a lot of what I'm talking about today is really due to Tom. Tom has really led the project which has developed retinal densitometry as a technique at Cardiff over the last 10 years. And he's worked very closely with UK Astronomy and Technology Centre in Edinburgh in terms of development, which I'll come to a little bit later on. And also, as we've just heard, Rhiannon Reynolds is also an ophthalmologist who is a supervisor on Vera's projects. So the top row, we're all involved supervising with Vera there. And as I say, I've got colleagues on the bottom, Jennifer Acton, Tony Redmond, Louise Terry, all academic staff members with their own projects. And also just Krishna Patni's, another of our PhD students, along with Turkey Alamashlok within the school there. So as I say, when you see this today, it is a team effort and we all contribute in our own ways. So moving on. Okay, so some of you may be familiar with this, but I'm just going to start off by really just giving a little bit of background of age-related macular degeneration, and particularly where it relates to the imaging densitometry that we're going to be talking about today. Okay, so probably just to start off really, age-related macular degeneration, as the name implies, is a condition that becomes more common with age. We know that there are certain risk factors that we have some control over, in terms of smoking and influences of diet and sunlight exposure. But really the biggest risk for it is age. So as we get older, that's the main thing there. But there is also strong genetic and family history components to the likelihood of you developing this condition. In terms of actually what happens there, obviously this is a condition which affects the macula being the sort of center part of the light sensitive retina, which covers the back of our eye. And the macula is the area where the light that enters the eye through the cornea, the clear front, travels through the lens and is then focused onto the retina. It's the point which corresponds to when we're looking straight ahead. So the macular region is the area where the light falls for when we're looking straight ahead. And this area is specialized with a very high density of light sensitive cells, which are called photoreceptors. And these allow us to see in detail for activities such as reading, for driving, for seeing faces. So it provides that detail. These cells here also are specialized for seeing color as well. And obviously in age-related macular degeneration, that area is affected. And as the condition advances, damage occurs here and ultimately can damage the function of those things, which unfortunately leads to vision loss. So what I'm just going to go through now is a little bit just focusing really about what is happening at the retina in the macula. So the slide I've got here, if you can't see it that well, it's not really, I'll try to explain it. So the key cell which we have in the retina in terms of vision is the photoreceptor. And essentially the photoreceptor could be thought of as like these small little tubes that are filled with light sensitive pigments, which we call visual pigments. Okay, and it's still, each little photoreceptor's little tube is filled with millions of these visual pigments. And why these are important is that these visual pigments are, when they're hit by light, they react to the light, they cause a chemical reaction, which causes the photoreceptor to respond and signals the presence of light. And that photoreceptor then transmits that as a neural impulse into the other cells in the retina, and that then is transmitted to the brain, and that allows us to perceive light. Okay, and so we have this whole layer within the retina of these photoreceptors, very dense, all very next to each other, each full of millions of pigments, of millions of these visual pigments. Okay, so this layer of photoreceptors sits on another important layer of cells called the retinal pigment epithelium, which sort of envelop and support the health of these photoreceptors. This retinal pigment epithelium has pigment in it, which where the name comes from, which absorbs stray light to help us see, but more importantly, it supports those photoreceptors. It, across this thing, it supplies the nutrients to support the photoreceptors. It removes the waste products. And as we will see a little bit later, it has an important role in regenerating those light sensitive visual pigments so that they can continue to perceive light. Beneath that is a membrane. So all these the RP cells sit on a membrane, and below that we have a very dense network of blood vessels, which sits underneath the RPE. And you can think of this as more like a plate of spaghetti. There's just hundreds of blood vessels with lots of blood traveling through them. And this is a very important structure because it provides the RPE and the photoreceptors with the energy and nutrients they need to work. And this is one of the most energy intensive parts of the body. The process of seeing and processing that into visual information is very energy intensive and requires lots of nutrients. So this typically these structures all work very well, okay, continuously working every day of your life when you're born, and continue as you get older. As you age, these processes, the transfer of all these nutrients and removal of waste products, gradually you start to build up, as a normal aging process, around the membranes, particularly between the RPE and the choroid, those blood vessels. We start to build up with lipids and fats, which build stock deposits. In age-related macular degeneration, one of the earliest signs is where these products aren't being cleared, and we start to get a build-up underneath these RPE cells. And these, you might be familiar with this, these are the drusens or yellow deposits that might be one of the earliest signs picked up when you go for your eye tests. Okay, so these waste products build up underneath the RPE. We also can get deposits building up above it, and these can occur between the photoreceptors and this RPE layer. At this early stage of AMD, although we have these deposits, typically vision remains unaffected. You can still see the small print, everything else, but we've got these deposits. So we know that something is not quite working, but typically the vision is reasonably okay there. There are some changes that occur. We do know that in these early stages, people have difficulty adapting to bright lights. So maybe going into a dark room from a bright sunny day, their eyes can take longer to adapt with that. But typically, that's there. So that's the early stage. Now, from this, we can then go to the more advanced AMD. And this is unfortunately where we start to get damage to the structures that results in vision loss. So this can either occur in one of two ways. So we can get a buildup of these deposits as these drusen become larger, more confluent, as we get more of these subdrusenoid deposits. The risk of progressing to the more advanced stage increases. And over time, we may develop atrophy where buildup of these products and the wear and tear that's happening in the retina leads to atrophy where the photoreceptors in these RPE cells go on to die. And as they die, they're unable to see, and therefore we start to get vision loss with that. And that tends to be a gradual process. And unfortunately, there's no effective treatment for that at this time, although there are research looking at stem cells to try to regrow the damaged cells that are lost. And there is also research which is looking at drugs and therapies that can try to prevent the atrophy developing with that, ideally preventing the loss of those cells to preserve the vision. And one of the things we're interested in the macular research group is trying to find ways to better detect the changes before we end up with that vision loss. So many of these treatments will go through clinical trials where they try a treatment against a placebo, but they'll have to monitor over time if those treatments are effective to see whether the atrophy develops. That can take several years and can make it quite slow to develop treatments. Whereas if we had some measure that maybe in this early stage, before we got to the atrophy, we could maybe detect whether a treatment's having a positive effect, maybe the retina's functioning better, or those deposits disappear. Those are sort of things we're interested in because they could help speed up the development of treatments if we can see they're effective before we end up with vision. So that's one of the things we're interested in. We'll see how retinal imaging densitometry might be able to help in that scenario. The other outcome of AMD is a neovascular outcome. So the build-up of these drusen, to build up these deposits, the difficulty in transporting oxygen and nutrients to the RPE and photoreceptors can lead to hypoxia or a lack of oxygen. And sometimes this can trigger new blood vessel growth. And unfortunately, these new blood vessels grow through from the choroid into the retina. They're leaky. They tend to leak, they bleed, they leak fluid, and that bleeding and fluid can damage the structures. And if it isn't treated quickly, that can lead to permanent structural damage and unfortunately vision loss. Thankfully, we have a number of treatments now for neovascular AMD. You might be familiar with the injections to the eye, the anti-VEGF drugs. So these target the growth factors, encourage that growth, causing those vessels to regress, hopefully removing the blood and fluid, which can prevent further damage to the structures. And if you can remove the fluid before too much damage occurs, you can often get an improvement in those ones. But it's critical for that treatment to be provided as soon as there's any detection of any new blood vessels. to get them to be treated. If it's left untreated, we get permanent loss, and that's where we lose vision. So that's just an overview of AMD. Now, to understand how densitometry works, we first need to understand a process called the visual cycle and how this is key to our vision. So I've tried to simplify in this slide. So what we're looking at here now is we're looking at a very magnified section of the retina at the back of the eye. So on your right-hand side of your screen, you can see some squiggly red lines there. There's a choroid, that dense network of blood vessels beneath the retina. We then come to the black line there, which is this membrane that sits underneath these RPE cells. And then we've got these hexagonal cells, which is the retinal pigment epithelium. So these are these cells that support, nourish, and maintain the photoreceptors. And this little image here, this is one of our photoreceptors, okay, divided into segments. But the area important is what we call the outer segment, which is a sort of purple here. And this is the tubular part, which is full of all these visual pigments. Okay. And so when we have light that enters the eye, okay, so we've got this little light here, this little sun. So we can imagine that light comes in, that enters the eye, and it hits the photoreceptor, and it actually reactivates these visual pigments. So with the light going in, the effect of that is that these visual pigments respond to the light, and they actually change color. They start off a slight purpley color, and then they become clear. And during that process, they trigger the photoreceptor to respond to the light, so it allows us to see, and the little nerve signal is sent. So as the light hits it, that becomes bleached. What now happens is that all of those bleached visual pigments are now insensitive to light, and they need to be recycled. They need to be created back to being light sensitive. And so they are transported out of the photoreceptor and to the RPE cells. And they undergo a series of chemical processes here to allow them to be made sensitive to light again, regenerating. And this requires oxygen and nutrition from the choroid, that process. And then once they've been regenerated, they have to be then transported back to the outer segment of the photoreceptor. And this process can take a long time for them to be recycled. But as it does so, the photoreceptor will fill up again with these regenerated visual pigments and be ready and sensitive again to light. And the next lot will hit it, they will bleach, and they'll have to go through this process again. So this regeneration of these visual pigments is called the visual cycle. It is very energy intensive, and it requires the visual pigments to transport from the photoreceptors to the RPE cells and then back again. Now, this is the process in typical eye. In age-related macular degeneration, as we've just seen, we start to get these deposits that occur between the RPE and the choroid, but we also occur between the photoreceptors and the RPE. And these, we believe, will slow down this process. Okay, and we believe this process slowed down because one of the effects of this bleaching and regeneration can be measured in a process called dark adaptation. Okay, so dark adaptation is our ability of the eyes to change sensitivity to light. And as I already mentioned, we know this is something that's affected in early AMD. So when the pigment is entirely regenerated, the eye is very sensitive. It doesn't take much light to hit it to be detected. Once it's all bleached, the light is going to pass through and not going to respond to. So we say we have low sensitivity. As the pigment starts to regenerate, as more visual pigment returns to the photoreceptor, the sensitivity increases as the pigment increases. And that process can take up to 40 minutes in a healthy eye. And once it's all regenerated, the eye is at its maximum sensitivity again. Okay. And we know that people with early AMD, that process of regeneration we measure from dark adaptation is slowed down. It means it takes longer for the eye to return to the sensitivity after it's been exposed to light. Okay, so we have this process, this visual cycle. We know it takes a while for that visual pigments to regenerate it, and we know that it can be slower in AMD. Okay, so what is imaging retinal densitometry, and how does it work? Okay, so to begin is just, I'm just going to tell you about what densitometry is. It is simply, it's simply a measure of absorption. So in a very simple thing, we can measure absorption by, we have a sample, we shine a light through it, and we compare the brightness of the light before and after. And if the light gets dimmer, it's absorbed by what you're passing it through. Okay, that causes a difference in the brightness from before and after. Okay, and we call this difference the optical density of what we're passing the light through. Okay, so if you think of if you shine a light through a mug of a squash or something, obviously it gets duller as the light passes through the squash. If you've got a concentrated amount of squash in the bottle, it's much darker. It's a much darker color because less of the light is passing through it. So essentially, densitometry is the measurement of how much light is lost as it passes through a material. Okay, why is this important? Well, because those photoreceptors, okay, we can use densitometry to measure how much visual pigment we have. The visual pigment, when it's regenerated and active, has a purple color, and we can measure that. Not easy to measure it, but we can measure it in a similar way. We shine light into the eye, it passes through the photoreceptor, it reflects back out of the eye. Some of that light is lost because it's absorbed by those visual pigments. So densitometry has been used to study these visual pigments in the eye in labs for many years, since the 1950s. But applying that to actually looking at eyes of real patients sat in a clinic has been a difficult challenge. So in terms of studies of densitometry in labs, we've had, they usually measure at a single point. They have one beam of light that goes into the eye and comes back, so we're only measuring one location. Often to keep the patient still, they have to use, in a lab, a dental impression to keep their heads in place. And the procedure is obviously not very applicable to using with patients in real life. So in terms of developing our imaging retinal densitometer, okay, what we want to be able to do is to look at those regeneration of visual pigments, and also to be able to do it over the whole macular area, not just at one point. So as a starting point to how this device works, we have essentially a camera, which you might encounter in a normal opticians for taking a photograph of the back of the eye. Okay, but what we've had to do is develop to overcome a number of problems for that. Firstly, patients' eyes move around a lot, so we have to control those eye movements. So we have to have cameras which are positioned to monitor the eyes, tracking the position of the eye, and the camera has to, and the entire optics within the camera needs to be able to move and keep following that eye, so that we know that the light that goes in is where it's coming back from. Okay, and to do that, we've worked with collaboration with the UK Astronomy Tech Centre based in Edinburgh, bringing together vision scientists and, in this case, astronomers who normally would work on space telescopes and satellites. But the solutions to their problems of looking at and detecting very small stars, millions of light years away, and be able to track it with their telescopes, can be used in the same way to allow us to track the position of an eye with our cameras. And so part of the technology we have used is to track the eye in that way. Another similar problem which you have with astronomy, which we also have in this circumstance of trying to detect the densitometry, is that the visual pigments make up a very small proportion of the retina, and in order to detect the density of the visual pigments within these photoreceptors, we are dealing with very, very small amounts of light, which may only be a few photons and may be very difficult to detect. So part of the densitometer actually uses a camera which would be normally used in space on telescopes used to detect very dim stars. We can detect even the very smallest amount of light with explicit detail. So this is built into our device. The final challenge is that in the human eye, we have actually different types of visual pigment. We actually have different types of photoreceptors. So there are rod photoreceptors, which are very common, which cover whole of the retina. These are generally sensitive to low light conditions. And we also have the cones, which are a slightly different sensitivity. These are the ones that are mostly found in the macula. They tend to deal with our high detailed vision. They also have the ability to detect color. Okay, and each of these have slightly different visual pigments, slightly different to detect. So how we pick these up is by using a special technique called multispectral imaging. Multispectral imaging simply is instead of taking a single photograph, what we do is we take a photograph at different colors. So essentially like taking a photograph where we only look at one color of the rainbow, and we then take multiple ones in sequence. And why we do this is that by taking lots of images at with these different wavelengths with different colors, we are able to get a picture of your retina, look at the differences in the colors, and we can, because we know the color of the visual pigments that we're trying to look for both rods and cones, which are slightly different, we can mathematically extract that information and tell exactly how much of each type of visual pigment that we have present. Okay, so I'm aware that's quite a lot of different complicated elements, but we have combined this space technology with a more traditional retinal camera in order that we can detect the small amount of light that comes back as light passes through these photoreceptors, able to work out which type of visual pigment we have, whether it's rod or cones, and we are able to track the eye so we know where it's coming from in the retina. And from that, we can actually identify what the amount of visual pigment that we have in the eye, and more importantly, how quickly it can regenerate. And I'll explain how that works on the next slide. Okay, so this image here just shows our device in practice with an operator and somebody sat at it. It's like many devices you may come across in practice. All of this advanced technology is all contained within that cover, and the patient simply sits on the chin rest and looks into it, and what they will look at a target whilst it takes the recording. So how does the ID work in practice? Well, for the participants, we start by using drops to dilate the eyes, like many people would have when they go to have their eyes examined at the hospital. And the first step is to adapt the eyes to the dark. What we want to do is to get all of the visual pigments in those photoreceptors to regenerate to the maximum amount. And so this normally takes about 20 minutes to happen. So the patient will sit in the dark with that. What we then do, once the eye's dark adapted, we move forward on the chin rest, line up the device, look at a target. And what the patient will see then is we'll make our first recording, and they'll see a target looking like on the screen, which simply presents a series of colors, like looking at a rainbow changing, and you just look at the target and these colors cycle through. And as it does so, it's taking images of the back of the eye, this multispectral imaging, lots of different colors. and then it mathematically can work out the amount of pigment that's present. So we record this signal in the dark. We then present a bright light to the patient. And what this does is then bleaches all those pigments. So they go from being purple, exposed to light, they respond to the light, they all become bleached. And we do this until for about 2 minutes, until almost all the visual pigment has disappeared. So there should be now no visual pigment present in the retina. We then record our multispectral again, and we compare the difference. So essentially how we work out how much pigment we have is by comparing the images in the dark when we had lots of pigment, and we now compare it to the afterwards where we should have no after the bleach where we should have no pigment. And simply we take one set of images from the other, and that tells us how much pigment we have in each of those photoreceptors in the retina. And because we have a photograph, we know this across the whole macula that we've imaged. The next step, so now we've now worked out how much visual pigment we have. What we then do is then we record this multispectral imaging for 10 minutes after this bleach. And as it does so, we keep repeating this sequence. And as the visual pigments recover, we are able to detect the small signal of those visual pigments as they regenerate. Okay. And so after about 10 minutes, we've seen it starting to recover, and we can work out the rate or how quickly it's regenerating. So once we've performed this procedure, at the end, it's all automatic processed by computers on our system, and we then it gets output as little plots of the density and the recovery rates as a series of images, which then can be interpreted by the clinician. Okay. And so what Vera is now going to go into is to talk about her her project, which uses this imaging densitometry, and how by understanding this optical density and the recovery rates, how we can use this to better understand AMD and the possibly as a way of detecting early changes in this condition.
Thanks, Ashley, and hi, everybody. Thank you so much for having us. I'm Vera, and I'm Ashley's PhD student, final year PhD student, and I'm just going to tell you a little bit about how I'm using imaging retinal densitometry in my project and what are the questions that I'm interested in trying to answer using this new technique. So as a research group, we are very interested in AMD, and more specifically, as Ashley mentioned, we're very interested in the early to intermediate stages of AMD where there hasn't been much vision lost. So in order to try to figure out what's happening with AMD using this new technique, before we had to study what is happening as we age, since, as you know, age is the main non-modifiable risk factor for this condition. So the first thing I'm going to be talking to you about is what we found about visual pigment as we age. And then I will tell you about what we are interested in studying with the AMD group of participants as well. So starting with our results from the age study. So in the next slide, I will be talking to you about optical density, so the amount of visual pigment that is in the photoreceptors, as well as recovery rates, which is what Ashley has just been talking to you about. And I'm going to be separating that into the two photoreceptor types. So we've got rods, which are the ones more responsible for seeing in dimmer conditions, and we've got cones, which are the ones responsible for really clear vision and color vision as well. So if I go to the next slide, it's not changing the slide. There we go. Right, so here I've got two graphs. I will just, if there's anyone with difficulty seeing the graphs, I will be explaining them. So on the left-hand side, we're looking at the rod optical density. So we're just talking about rods for now. So on the left-hand side is the total amount of rod visual pigment that we have. So as you can see, there is a positive relationship with age. So as we age, we actually have more rod visual pigment, which was quite surprising to us because we tend to think of aging as everything declining and everything decreasing, and we actually see the amount of rod visual pigment increasing. On the right-hand side, we have the recovery rates. So after we shine the bright light into the eye, how quickly does the eye recover from that bright light? So here, interestingly, again, we didn't actually find a relationship with age, but this graph is actually very, very interesting, and we're going to be looking a little bit more closely at it. So if I go over to the next slide, so we have more visual pigment in rods, like I said, and we actually see the tendency for, as we age, for that regeneration to also be faster. However, for the people that are aged over 60 years old, we see a big difference in recovery rates. So for some people, they recover really, really fast. And for for a smaller group of people, we see a slower recovery. So we see a deficit in how their retinas are working. So I've highlighted those in this yellow circle here. So that's a group of people that, unlike all the others, have a much slower recovery. Now, this is a group of people with healthy retinas. So we've done all the different types of photographs to make sure that we can't see any changes. So these are healthy retinas. However, there is this small group that is already showing a change in function. So if you remember what Ashley explained earlier, in dark adaptation, we know that dark adaptation is affected from a really, really early stage in AMD. In fact, it can come even before there are signs of AMD in the pictures that we take. We can already see a deficit in dark adaptation. And basically, visual pigment regeneration is the underlying mechanism behind dark adaptation. So we would expect that this group of people perhaps would go on to develop changes in their macula and perhaps go and develop age-related macular degeneration. There is no way we can tell that right now. The only way to do that would be if we follow these participants over time, which is something that we look to do in our future research. It would be really interesting to follow them and see whether they go on to develop age-related macular degeneration. I'm going to show you now a few examples of what the data actually looks like. So what comes out of the densitometer. So if I go over to the next slide, here I've got an example of a 39-year-old and a 73-year-old. So big difference in age. However, you can see that there's not many differences between what you've been showing at the top and at the bottom. So on the left-hand side, we have the amount of visual pigment, and there is a slightly increased amount for the 73-year-old. When we look at the recovery, it's really hard to tell any differences. They're quite similar in terms of how quickly the pigment is regenerating. But if you remember, I told you that there is a small group that showed a delay in recovery rates. So if I go over to the next slide, there's actually a participant that was younger than the 73-year-old. So in this case was a 63-year-old. And you clearly can see difference between the plots now. So the red that you see means more visual pigment or faster recovery. And you can clearly see that the 63-year-old here is showing a deficit both in how much visual pigment they have for the rods and how quickly those rods are working. So even though it's a younger person, it is showing a deficit. Right, moving forward to what's happening with the cones. So the different photoreceptor type. Here, one of the main points we conclude from this is that cones and rods don't age the same. So we don't see the same thing happening with one photoreceptor type than the other. In case of the cones, the amount of visual pigment actually doesn't change much with age at all. So we didn't find a relationship with age with the amount. But in terms of the recovery, so how well that this part of the retina is working, it decreases as we age. So there is no specific group here that decreases more than the other. It seems to just decrease for everybody with age. And if we look at the same examples as earlier, but now looking at what's happening with the cones on the next slide. So as you saw with the rods, the red was around the periphery of the macula, while the cones are concentrated in the middle. So now we see a peak at the middle. So for the 39-year-old and the 73-year-old, you can now see a big difference. And there is, in fact, a decrease in the function for the cones as we age. And if I go to the next slide and look at the same 63-year-old participant, in this case, their cone recovery rate actually matches what would be expected for the age, unlike what was happening to the rod recovery rate. So if I go over to the next slide, I actually presented these results at a conference earlier this year. So I went to Seattle to ARVO, which is one of the biggest ophthalmology conferences in the world. So it was a great pleasure to share these results with the scientific community there. So very quickly going on to the AMD study now. Yep, going on to the next slide, just showing you an example of what the imaging retinal densitometry results look like for someone with AMD. So we can see the two aging ones on the left and then the intermediate AMD on the right. So just as clarification, intermediate AMD is still a stage where it hasn't developed to advanced, so vision is still conserved. But as you can see, it is very clear that there has been changes, especially in the recovery rates of this participant with macular degeneration. And if I go over to the next slide, in my study particularly, we are very interested in studying the subretinal drusenoid deposits, which is this type of drusen that accumulates above the RPE, so really close to the photoreceptors. This kind of deposit is also associated with more chances of developing late-stage AMD. And then the other aspect we're really interested in studying is areas where there's a lack of blood flow. So Ashley told you a little bit about this layer of blood vessels that is underneath the photoreceptors and the RPE. So we are interested in studying the areas where there are changes in the blood flow and seeing how that can affect the visual pigment. So to do all this, if I go over to the next slide, besides imaging retinal densitometry, we use many other techniques. Some of them will be more familiar to you than others. For example, we've got an example here of an OCT. So you get this 3D vision of the back of the eye. So these are all examples of a patient that has macular degeneration. We also do color photography. And then perhaps less common techniques such as fundus autofluorescence and infrared imaging. We use all these techniques so that we can make sure to differentiate between those two types of drusen that I was telling you about earlier. The other technique we use is called an OCT angiography. So you might have never heard of this technique before, even though it has OCT in the name. It's not exactly the same thing. So the OCT angiography actually looks at the blood flow. So the picture you see here, the areas that are brighter means that there is blood flow in those areas. And if you look closely, there are some areas of darkness in this picture. And those areas mean that there is either a really slow blood flow or no blood flow at all. And we can then compare these images. We can overlap them with the densitometry map, which you can see here on the bottom right corner. And then we can study what is happening in terms of the structure and what is happening in terms of the function. And we can do it point by point basis, which is something that was not possible before we had this technique. So yeah, I'm getting to the end of my PhD project and now onto analysis of the AMD study. I just quickly wanted to tell you that at Cardiff University, we're also starting a clinical trial looking at a new treatment for early to intermediate AMD. So it's something else I'm working on and is quite exciting. I really look forward to sharing the results from the AMD study as well as our findings from the clinical trial in the future. Thank you so much for listening. In the last slide, we'll leave you with our contact details. So there is the Macular Research Group email address and a phone number as well if you prefer to give us a ring. And there's Ashley and I's email addresses separately if you wish to contact us directly. Thank you so much. I look forward to hearing your questions. Yeah.
Brilliant. Thank you both so much.
Yeah,
Ashley, I'll
thank
get you to unshare if that's okay, so everybody can
Yep.
see everybody's faces.
There we
So
go.
I think one of my reflections, having been here a while, is the lack of being able to tell who is going to get AMD and then who is going to be wet, who is going to be dry, who is going to have a. So is your hope that, because this is giving us some data, isn't it, in terms of that comparison between people who don't get AMD and people that do, that this could then give us some insight into who is more likely to develop AMD and the different types of AMD than we know so little now?
Yes, this is the hope. So, I mean, one of the the results there that Vera sent are quite exciting in that regard, because we were looking at the elderly patients, but with those with completely healthy retinas. So we had no reason to expect that any of them had AMD. They had no signs of it. They wouldn't be diagnosed with it. But as you saw from those results there, once we get to over 55, there's a clear distinction. Most people had a pretty quick recovery, and we had that small group. Now, because that was taken at one point in time, we have no idea whether those people are likely to go on to develop AMD. But it does, our hypothesis would be that they have a slow rate of recovery there. We know that in early AMD, they also have a slow rate of recovery. So could this be an indication that these are people at risk of then developing it? Of course, we'd only find out by following up in later years to see whether that develops. But it is an interesting hope from that.
I would just add also, you did mention that it would be interesting to differentiate between the people who go and develop dry advanced AMD or wet advanced AMD. And I think looking at our AMD results, at least from what we've seen so far, we've not finished our analysis, but not everyone looks the same in terms of their the pattern of loss of recovery. So I'm hoping that there will be a clear distinction between the AMD groups that perhaps, again, looking at them over time will tell us a little bit more about what a group is more likely to go and develop than the other.
It was very striking, that image of the rods for somebody with to be able to actually see it there felt quite exciting and felt like something that we just haven't had up till now. So I think, it's so great to see that, the dream would be, wouldn't it, that you'd be able to tell somebody you are more likely to, we can see at 50, 60, you're more likely, these are the things that you can do now that might mean that actually you wouldn't then need anti-VEGF, or would it be that actually you might start that earlier if we've got that data. So yeah, very exciting. So there's some excellent questions in the chat. Gillian asked that the scan that you're doing, it's the same scan, presumably you've got the machine, and the scan that you do to do the dark recovery is then the same as the second one that you...
Yes, so it's the same method, it's the same, uses all the same technology. So it's just a process of steps. So this optical density that we're measuring, which is essentially the difference between when we have all the visual pigment there in the dark, i.e. when it's all fully recovered, and when it's all gone, when it's bleached. And so the way we measure it is simply we dark adapt the eye so we have all the pigment there. We do our image, we pick out those pigments using that multispectral imaging. We then bleach the eye, and we know then once all that light goes in there, all that visual pigment goes from purple to clear. We then do the visual, the multispectral imaging again, and it's simply the difference. One take away the other. The difference between the two is tells us the amount of visual pigment. It's a little bit more complicated because we've got those different types of visual pigment, which is what we talked about in terms of that multispectral imaging. But simply, it's one image take away the other, and the difference is the amount of pigment we've got.
It's amazing. Ramesh says, are there more mitochondria in the cone photoreceptors? And if there is, why do the mitochondria not protect against AMD? Which you may not have the answer to, actually, to be fair.
I don't necessarily, although I do know that there's lots of research into the role of mitochondria in AMD. One of the things is that basically the energy efficiency. So mitochondria are essentially the power batteries that power all our cells in our body, and there is absolutely tons of them in the outer retina, because the outer retina has a huge metabolism. This process of seeing, regenerating these visual pigments and all the mechanics around them takes an awful amount of energy, which is why it needs all that blood supply, all that oxygen. And we do know that as we age, mitochondria become less efficient and less productive. So as a lot of the theories of the development of AMD are around essentially the mitochondria becoming less effective. And there are studies that are looking at one, in trying to explain AMD, what's happening in the mitochondria, looking at the genetic things, how they're functioning. But there's also some studies which are actually looking at ways maybe increasing the function of those mitochondria so that they can produce more energy or go back to the efficiency you might have had when you're younger, and hoping that by doing that you might be able to offset some of the negative impacts and possibly prevent or slow progression of the condition.
It's not great aging, is it? We had somebody who was an expert in aging at our conference talking about, you know, all of the ways that aging impacts on the body, and AMD being just a part of that. So, but yeah, how do we tell who is more likely to get AMD versus those that don't? And what is that difference? What is it about that person as a whole and their eye and what's going on in the eye that means actually I may not get it, but Ed might? You know, what is that difference? Gillian says, I've just been referred for a scan for PED and it starts with pigment. How is that linked to AMD?
Okay, so a PED is a pigment epithelial detachment. So what happens in some cases is that the layer which we talked about, the pigment epithelium, sits on another layer called Brooks membrane, and underneath that is those choroidal blood vessels that supply the oxygen. So in a PED, as it says, it detaches, so the pigment layer separates. There could be different reasons for why it separates. Sometimes you get a large buildup of those drusenoid deposits, the pigment lipids, and over time that can cause that to separate. So that's called a drusenoid pigment epithelial detachment. Sometimes it can occur due to the buildup of fluid or new blood vessel growth underneath from the choroid through, which causes it to separate. So there are different causes of PEDs, but essentially, as the name suggests, it's a separation. Pigment epithelium separates from the membrane beneath it.
Karen's asked a question that we get asked a lot, which is, you know, she's got an 89-year-old mother who has wet AMD. We know the genetic link. What can you do? Our webinar next month is going to be about the update to the AREDS 2 supplements. So join us this time next month and we'll give you some hints and tips as to what you can do. So how many, out of curiosity, how many people are in your study at the moment, and how often are you running those scans to look at, because as you said, Ashley, there's something, isn't there, about actually what's the length of time we need to be following these people to see some of these people that you've seen that have this anomaly, what is going to happen to them? You know, presumably that could be a 20-year study. So how do we do that work so we can then get that insight that then gives us the data that says, right, actually we need to do these scans at this point and this is how frequently we need to do them? So
I mean, it's a very good question. I say one of the limitations always in terms of AMD research is the time.
Yeah.
And so many of the, so a lot of the studies that we do for cost reasons tend to be what we call cross-sectional. So we look at people at a certain stage, we look at people without it and compare the two. But actually, you know, as I mentioned earlier, one of the challenges in terms of developing treatments is that we basically have to administer a treatment, we think this is going to work, but the only way we really find out if it does work is whether those people taking that down the line develop atrophy or develop neovascular AMD. So that's not a very efficient thing, but it is quite expensive, because if you need to get a study, you need to see them at baseline, you need to get all the details, you need to do all the scans. You know, for example, if somebody with an early stage we're interested in, you know, we need to do that. But in order to find out what actually happens to them, we have to bring them back in 2 or 5 years' time to see what's happened. We have some idea of what might happen from other studies, but until you've actually got those in. So that is always a challenge. In terms of numbers, I think we had about 60 or so. I can't remember the exact number, but it's around 60 in the age group there, which you can probably see with all the dots on the graphs. And we've got a similar number aiming for the AMD participants. So yeah, so there's a lot of people, but it takes
yeah. Oh yeah. Yeah. And what's your hope that comes next for this study? You know, where do you hope it takes us?
Well, in terms of Vera's project in particular, well, see, the analysis we're looking at is actually, you know, how does the densitometry relate to these individual signs, particularly blood flow dropout and those pseudodrusen we talked about. And the reason why we're looking at those is, and particularly the subretinal, sometimes called pseudodrusen, they have 2 names, is that they are often associated with higher advancement to advanced AMD, but in particular, they have a particularly strong association with reduced dark adaptation. It's a dark adaptation issue. So they were a particular target for that reason, for us to look at. And so hopefully we can look at that. And what we also want to see is those ones, as we've already mentioned, following up, hopefully in the future, those with poor recovery rates, but are otherwise normal, that we think are healthy, but have those rates, because that would be really interesting. The densitometer more generally, though, we've got several projects which are looking at using that, and including the Samadhi trial that we just had mentioned there, which is looking at a potential treatment. We're going to be using the densitometry as part of that. And one of the reasons being is that with any treatment, as I said, following up people for a long time, we're often just waiting to see what the outcome is. So what we're hoping to do is if we collect the densitometry with that study, that we might learn more about how the densitometry results relate to the effect of that intervention or not. So and potentially what we'd hope to be is maybe have the densitometry in the future being used instead of waiting to see somebody who develops atrophy or neovascular AMD. We can go, well, we've got dark adaptation, we've got slower recovered rates and poor dark adaptation. We give them, say, a drug. If it makes that better, it says that the retina is functioning better, we can maybe use that as a way to see whether the drugs are effective before having to wait 5 years to see whether it leads to atrophy or neovascular AMD. So the hope would be is that these metrics that we're generating might have a value in actually telling us about the function and telling us at a much earlier stage than waiting for vision loss to occur. So that's our hope, but we are unfortunately a number of years away, and lots of research effort from that point.
Presumably the dream would be that as part of going and having your sight test, you know, we know now that we're telling everybody to have an OCT scan, that the ultimate hope would be that you'd have this as part of your routine eye test, and it would give you, you know, something that would tell you, okay, I know, you know, as you said, that you can see these changes and the person has still got vision. There's nothing. So actually, having that knowledge much earlier could then mean, there's a treatment that we could do here that will mean you never lost your sight, that you may have a diagnosis as such, but actually we've intervened much earlier as a result of a test that you had in an optician, and you know, you don't need to go through all of the stuff that we know our audience go through. So that's the hope, isn't it, and the dream.
The ideal thing is that you prevent actually people getting vision loss in the first place, that you can one, detect people early enough and you can intervene with a treatment before there's any vision loss, before there's any effect whatsoever. That's where we want to get to. And we're hoping that densitometry will be one of the, we've obviously got OCT and imaging and stuff, but that densitometry will add another element to that. And that because it looks at the function, because obviously a lot of the other things just look at the structure, this looks at how the things are functioning, that extra bit of information will really help us, you know, look at what's happening in the retina. If we see that functioning starting to decline, maybe we can stop it declining, but by detecting that decline also, we might be able to pick it up that bit earlier. That gives us more options and hopefully better outcomes.
Yeah. Oh, well, we'll keep our fingers crossed. Thank you both so much. It's you know, our audience are just, we all want to know that things are moving forward, that there's hope, that there. So it's great. It's so great. So thank you so much for joining us, both of you.
Thank you. It's been absolutely fascinating, and to hear how this is, you know, giving us a different way of looking what's going on in the eye, and as you say, helping to test treatments and hopefully pick people up earlier so we can start treating this. So thank you, really, really fascinating, and thank you everyone for your questions. Hopefully we got through most of them. So as Emma said, next month, please do join us. We'll be talking to Tianan Keenan from the National Eye Institute, and they'll be discussing the recent findings in the AREDS2 research, which is showing that taking supplements can also slow down the development of late-stage AMD, also known as geographic atrophy. And previously, those supplements were only recommended for people with the earlier stages of AMD. So it's potentially a really exciting discovery, which we look forward to hearing more about. Can you remind me of the date of that, Emma?
It is always the third Tuesday of the month.
Third Tuesday.
So it will be the 19th of November.
The 19th of November at 7 o'clock. So I look forward to hopefully seeing lots of you join us there. So thank you again, Dr. Wood and Vera. Really lovely to hear from you all. Thanks a lot. Good night.
Thank you for having us. Thank you.
Bye.